So in this video, I want to talk about a biochemical technique called site-directed mutagenesis. And we call this experiment also a Q5. And the Q5 is just the name from the company who sells you the kit and all the reagents necessary to do a site-directed mutagenesis. They call it a Q5. And so the company that does this, it's New England Biolabs. So you could go to their website and they have videos on how to do site-directed mutagenesis as well. But so I want to go through the basics with you, and the purpose here is, let's say you have a plasmid, or it'll be drawn plasmid there, and we have a particular gene in that plasmid, and that gene encodes for a protein that you're studying. And you want to understand the function of a protein. And so one way to understand a function of a protein is to understand which amino acids in the protein are essential for its function. And so you could have a hypothesis that say, hey, I think this lysine amino acid is really critical to its function. So let's exchange it for an alanine and see what happens to the function of the protein. And so you can do this experiment to learn about protein function. So if you want to see if an amino acid is important, you can do what's called a substitution, what I just described. Oh, there's a lysine at, let's say, position 20. Let's replace it with an alanine and see what happens to the function of the protein. Okay, so that's a substitution when you're just replacing an amino acid. Sometimes you need to be able to look at your gene and take out certain amino acids that you don't want anymore. And that would be called a deletion. And then there are times where you have your gene and you need to add in some, or the protein of interest, you need to add in some amino acids. So I want to take a look at those three versions there of what's going on. Okay? And so when we take a look at let's take a look at substitution. Okay. So substitutions with the Q5. What's happening is, let's just zoom in a little bit here into our plasmid. Okay. And let's say in our plasmid here, this is our gene of interest. Right here, there's our gene of interest. Okay. And we want to do a substitution, so that lysine to alanine thing. Okay. So we're going to have like a junction point, okay, like this. And this blue line that I've just drawn, it will make more sense momentarily. I'm just setting up the problem here. So what's going to happen is you're going to design a primer, okay, in a substitution. And what's going to happen is you're going to start on this blue line right here and design a primer that just goes in that direction. And that would be your forward primer. And within that primer, you have a codon that encodes for lysine, the lysine that's naturally in this gene. And so that codon that encodes for lysine, what we're going to do is we're going to change it to the codon that encodes for alanine. And then we're going to take our reverse primer here and go in that direction right there. So that's our reverse primer. So when we take and we zoom in, so what we're going to do now is we're not going to zoom into the whole gene. We're just going to zoom in just a little bit of the gene and write its sequence, okay? So what we have here, let's use this color here. Okay, so we have our 5' end, and I'm going to type in the codons like this. We have TCC, that's our 3'. And then we're going to have our 3'. So our 5' part, or this top strand right here, is going to be the coding strand. And the 3' strand here is going to be our non-coding. And so that would have to be not that. There. So that is the gene that I've just zoomed in right here, okay? Now, so this gene right here, let's see what it encodes for. These codons encode for a serine like this. Right there. Okay. So let's say in this particular example, what I want to do is I want to replace this aspartic acid right here. I want to replace that aspartic acid and turn it into a leucine in my particular example. I know just moments ago I said let's take lysine and replace it with, or substitute it with an alanine. That's a thing to do. But in my notes here, it looks like I did an example where I wanted to do this substitution. I want aspartic acid to be replaced with the leucine, okay? So we identify in the gene the codon that encodes for that aspartic acid. And that's the one that we want to turn into leucine, right? That's what we want to do. So what we have to do now is find that junction point. So what we want to do is this blue line right here, okay? Now what I'm going to do is give you some general principles here that, well, let's just do it real quick, okay? So there's the change. So this blue line right here is going to be, I'm going to put it right there. Okay. And so this forward primer now that I want to design, okay, I'm going to put it, I'm going to follow the diagram here. So this forward primer right here is on the top. So what that means is that the forward primer is going to look like this: ATG, GAT, GAT, and TTA. Okay, so there's my primer in the 5' to the 3' direction. But I made a mistake here. That primer right there encodes, GAT encodes for the aspartic acid. So what I need to do is replace that codon right there, okay, with what encodes for the leucine. And that should be TTA. So that codon right there is the asterisk in this little diagram right here. Okay? And so that is the forward primer. But remember, if you go to my video about PCR, this forward primer, where is it going to anneal when you take this DNA and denature it? Where is it going to anneal? That primer that I've just drawn right here in blue, it will anneal right here. Okay? And so that's the 5 prime, the 3 prime, and so it's going to synthesize in that direction. So by definition, the forward primer is the one that's going to anneal to the non-coding strand, or the template strand. And that's what we see. So this represents this, and we know the forward primer is going to anneal to the template strand, right there. So the reverse primer right here, okay, the reverse one, the sequence for the reverse primer is going to be this right here. CGT, GTT, and AGG. And how's that written? That's the 5 prime end, that's the 3 prime end, right? So that's the reverse primer, represented with this right here. But where does that reverse primer anneal? That reverse primer is going to anneal to the coding strand. Do you see how that's complementary? G to C, C to G. So this strand, or this primer right here that I have in green, is going to anneal right here. And we synthesize DNA from the 5 prime end to the 3 prime end. So it's going to go in that direction. And that would be our reverse primer. And this one's our forward primer. Okay? So that's how you design your primers to do this experiment. Now, the way that I showed you how to do this is mostly correct. And what's not correct about it is the size of the primer. You see how I only have like 9 nucleotides? These primers need to be much larger in order for them to anneal appropriately. So I just chose to do a smaller primer just due to space on the board. But when you go to this website, they will talk to you about techniques about how big the primer should be. But for the purposes of my class, I'm just more concerned that you could design a hypothetical primer that's small, because it's just assessing your understanding of what to do. And then if you do this in a lab and you really have to do this in real life, then you would, make your primers much larger and follow those techniques. So that's what I want you guys to know. So now let's move on to a deletion and see how that works. So for a deletion, let's say we want to delete those 2 amino acids out of our protein. So if we zoom in our plasmid here, and there's our gene of interest, okay? We need to look at that gene. Where are those A and M's? Okay? Where's our alanine and methionine? Once we identify and find where those are at, we're going to make a cut on both sides of the 2 amino acids that we want to remove. And so it kind of looks like this on the figure. Okay? And then how you design your primer is you're going to design your forward primer from that cut, and then the reverse primer on the other line. And so if we just do that based off of this little diagram here, and so this little inside here would be our A and our M. And so the primers that we would have to design would just go on this line right here. So our forward primer would be GAT, GAT, TTA, like that. That's our 5' end of the primer, 3' end. That's our forward primer, but where's that going to anneal? Right here on the template strand. And so that will go in the 5' to 3' direction, so that makes that the forward primer. And then what would the code be for this guy? It would have to be, the reverse would be our, we'll just put it right here, I guess. GTT AGG. So that's our 5' end, that's our 3' end. And that's the reverse primer right here. And that reverse primer is going to anneal where? On the coding strand. Just like that. And then you're going to, when you do that, you're going to get your linear forms of DNA. And then you just PCR it. And you get many, many copies of your gene that does not contain this piece. So what's going to happen is after you do the PCR reaction, your circular plasmid is now going to be a linear piece of DNA, right? So what's going to happen is you're going to have this whole piece right here, okay? And let's just see, we'll have this piece right here. We're going to have a little bit, where did that marker go? We're going to have this little piece, which is from right there. And then we're going to have this piece right here. And so this piece right here is right there. And what was in the middle here was the AM. But now that has been chopped out because we have made copies and multiple copies of this linear form of DNA, and this piece has not been copied. And so then you would take this linear DNA and treat it with your DNA ligase, and then you're going to put these back together. And when you ligate your linear DNA to form your circular plasmid, now you have the gene of interest in the plasmid minus this part. Okay? So that's what's happening in the PCR reaction. So now the last one that I want to talk about would be the insertion. So let's take a look at an insertion version here. So an insertion here would be, hey, what if we wanted to put a leucine in between alanine and methionine? What if we want to make that happen? So at that insertion point, what you're going to do is draw your line right there. So when we take a look and zoom in on our plasmid here, and we have our gene. Okay. And we want to insert it at that point. Okay. So what we have on this side right here is our alanine and then our methionine, and we want to stick leucine right in the middle there. What's going to have to happen is you're going to need to design a primer in such a way that your forward primer is going to start on that blue line right there. You're going to design your forward primer. Okay? But on that primer, you have to put the codon in that encodes for the leucine. And that's just going to be represented by the orange right there. So what's going to happen is on the first round of PCR, this little piece of fluff right here is not going to anneal to the DNA. It's just going to hang out. And then your reverse primer is rather straightforward because it just goes from this point of that blue line and go like that. So that's our reverse primer. Okay? So when we come to design the primer, let's start with maybe the reverse primer. That will be the easier one. So the reverse primer is just right at that junction, which is right here. So the primer would be GCA. All right? No. It would be GCGT, GTT, and AGG. And that would have to be the 5' end of the primer, 3'. We synthesize in the 5' to the 3' direction. So this reverse primer, where is it going to anneal? It's going to anneal right here. Okay? Now the forward primer, let's start with just the green part right here. So what would the forward primer look like? ATG, GAT, and let's just, we'll leave it at 9 nucleotides. So that's our 5' end of the primer, 3' end of the primer. Okay, and that is going to what? Anneal to the template strand right here, and then synthesize in that direction. Now it's the orange part here. Okay, that orange part, the little fluff that cannot anneal to these nucleotides right here, it's just going to hang off, has to encode for the substitution, for leucine. And the codon that codes for that is a TTA. So your primer is the orange and the green. It's that whole thing. Okay? And so what's going to happen now is that after the first round, let's draw what we're going to see, okay? After the first round of PCR. So after our first round of PCR, what's going to happen is we're going to have our linear DNA. And so this part right here, okay, this forward primer, the way I've designed it is it's part of the gene, right? So in theory, we could say this whole forward primer is just pink because it's going to match the pink DNA. And so this little pink primer right here, the forward primer, now that I'm drawing it in pink, is going to anneal to the gene. And so there we have it. Okay, so here's the pink part of our gene right there. Okay. And then we continue around, that's the rest of the primer, the plasmid. And then we come to this part right here, which is right there. Okay. So there's our gene of interest, right there. Now, I don't know if this is coloring very well for you guys, but this right here, the TTA, and this little squiggle right there, that's in orange. And so that is the insertion right there. That should be color. I don't know if it's contrasted well enough on your guys' end, but this is the orange part, and there's the gene. And so that's what we have. We have this overhang right here at this little DNA right there. And so then what we do when we do another round of PCR, what's going to happen is that is going to be part of the gene of interest now. And so you are going to be able to just have a linear form of DNA that now has incorporated this piece. So this piece plus what was originally there, I'm going to now represent just with that pink gene. And then this piece just stays the same, like there. So after the second round, you're going to start getting your plasmid there with the insertion included, not just fluffing and hanging off. It's going to be incorporated. And then you would take this version after doing 25 to 30 cycles and do the DNA ligase and seal that up. And you would have your complete gene right there with the insertion included, which was approximately right there. So that's how you would design an insertion. All right? So as always, if you guys have any questions or concerns, please reach out. I'll be more than happy to help.